Okay, so today we're going to be doing a gram stain. Remember last week we fixed a smear. So in order to start the gram stain, you need to have a fixed smear. We make the smear by adding 1 drop of water, a little bit of a colony, swirl it onto the microscope slide and allow it to either naturally evaporate or gently add it to the Bunsen burner and apply some gentle heat. So now we are going to begin this differential stain. The first step is to add crystal violet. We're going to make use of this metal apparatus that can keep our work area cleaner, and we're adding the crystal violet dropwise until it covers the entire smear. We then wait 30 seconds. After 30 seconds, we can wash the crystal violet away. And next, we will add the iodine. Gram's iodine is going to turn the crystal violet molecule into these large crystals. We'll add this to the top. You can flood the entire area to make sure the entire smear is covered. And we apply the Gram's iodine for 15 seconds. Of course, my tutorial is a little shorter. After 15 seconds, we can get rid of the Gram's iodine, do a quick wash with water. And our next step is to use the decolorizer. The decolorizer is almost always isopropanol. It's going to pop holes in the thin gram-negative bacterial cell wall, but let's see, I'm going to add it to the top for 30 seconds after I give a quick rinse. And it is not going to be able to penetrate the cell wall of the thick gram-positive microorganisms. So the crystal violet is going to remain trapped in the gram-positive organisms, and they're going to remain purple, where the gram-negative organisms are going to have holes popped in the thin peptidoglycan cell wall, and the purple color is going to wash away. After 30 seconds, we can dump the remaining ethanol, do a quick wash with water, and now we have our final step, which is the counter stain. Called the counter stain because it is the second and final stain of this process. Again, we can flood the entire area, and we're conducting another 30-second incubation. Now in this case, the gram-negative organisms are going to be clear because the decolorizer popped holes in it. It allowed the large crystal violet to run out, and so now we have these ghost-like cells that are gram-negative cells, and this pink dye is going to be applied to those ghost-like cells, and they're going to appear pink. So gram-negative organisms are going to look pink after a gram stain. Alternatively, our gram-positive organisms could not be penetrated by the decolorizing reagent. The crystal violet remained trapped inside of the cells, and now those purple cells are having a pink color applied to them, which keeps them a deep purple color. After that 30-second incubation, we'll rinse, even rinse the backside, making sure to get rid of any dye that is not associated with the smear. You can blot this dry with the bibulous paper and then begin to visualize your microorganisms under the microscope.